our synthetic biology project, and we're in collaboration with BE 755. And to my left is Christine, who we worked very closely with to motivate our project in the right direction.
Hi, I'm Christine. I'm a third-year BME student, and I'm in the 755 class, and my research involves studying the role of linked RNAs, and those are long non-coding RNAs. And to do that, I use fluorescence in situ hybridization, or FISH. And this is basically a method that uses 20 nucleotide long oligomers to hybridize to the target link RNA, and each of those oligomers has a fluorescent dye on them. That dye allows you to individually image single molecules of RNA. So for each of these sets of probes or oligomers I design, I need at least 48 of them, and they need to be individual and not align anywhere else in the genome to give off-target effects. So to do this, I've had to take a lot of time to actually not only design the probes through software, but then check that they don't align in other regions of the genome. And because of some of the limitations of tools like BLAST, you can't do that very succinctly. You actually have to extend the nucleotides longer so that you can ensure that there's no off-target binding. So this project is looking to find a quicker way to be able to do the work I do that typically can take me up to a day or a day and a half to do. It'll make it much simpler.
And next we'll take a look at what the original way to run these sequences are and our new enhanced method.
So let's take a look at what the current process entails using the BLAT system found at the UCSC Genome Browser. Here's the main screen. We find a box that you can input your sequence. I have a list of sequences received from Christine as potential options for her FISH probes. If I place a sequence in here and select the genome and assembly that she's been using, which is the MM10 found on the mouse, and submit that into the system, it returns a set of data and information finding the sequence on one DNA strand. I can copy that into a document, and actually, looking at the details, you can find the DNA surrounding that sequence and where that was actually found. The one in color is the sequence that we entered, and the surrounding sequences are found around that. Her next step is usually to copy the DNA in this sequence and set it aside while going back to the search engine. Here, she then retests the sequence with a plus and minus 10 to produce some additional information whether or not the sequence will be good to use. After copying the plus or minus 10, we run it through our system once again and get some information about this new set of 40 and whether it is considered a good set to use. Finally, she runs it 2 more times with 2 additional sets of sequences, plus and minus 5 and plus and minus 2. So we'll run the plus and minus 5 next, copy that, and now go back and run the plus and minus 2. Here you can tell that this method of performing all these tests on one sequence takes a while and can actually be prone to human error. If she wants to continue on to her next sequence, she would have to go back, copy the next sequence from the list, and start going through the same process again. You can tell that this gets very tiresome and long and repetitive, especially when you have a list of almost 100 different sequences and need to find 48 that are good matches. This is why we decided that our main focus on our project would be to improve the amount of time it takes to run all these different sequences through the BLAT system, as well as to automate the process of the expansions per sequence and running it through the system as well. In addition to that, since we are creating an API on top of this BLAT interface, we wanted to be able to save the data easily and clearly for future use, as well as easy reporting in applications such as OWL or just basic reporting mechanics with Word or Excel. So let's take a look at our design and how it's different from the basic BLAT search genome. Most of it looks the same. We have drop-down options just like before, which can let you decide which genome we want to be using as well as which assembly version. Here, if I take the same sequence in question that we used in the prior example and send it in, I can then actually input choices on what kind of expansions I want to use for this sequence and run all 4 in a row. Since she does each sequence of 20 with a plus or minus 10, plus or minus 5, and plus or minus 2, that's what I input. Now we can let it run. It'll perform all 4 tests in a row and return back a nice grid of all the information together. Each test is run and saved individually as well as the sequence that was run through the BLAT interface. The data is similar to what we got before, however, since it's in a tabular format, you can copy it easily to something like Excel. In addition to running one sequence at a time, our system can actually run multiple sequences as well as expansions all at once. So, for example, let's take 50 of the sequences in question and place them into our system all together. All 50 are here. We'll run it through, let's just do 2 expansions for now, and I'll actually time how long it takes to run the 50. In total, that will be 100 sequences run through the set, and we'll return once that's complete. In the meantime, let's look at how our application is developed and structured. We first take our input sequences and store them into individual BLAT jobs, which then can be run through the UCSC genome browser. We parse that information with JSoup and either create new BLAT jobs to run back through the browser or save them in our output. Using the browser version of our application, it will save and display the output in a nice, easy to read grid, while if you use the jar version of our application, that output will be saved to a file which then can be read using Node.js and incorporated as a JSON file for something such as OWL 2.0 or any other application which can use our standalone jar. And there, the system has just finished processing in almost 5 minutes and 45 seconds. Here we can take the data that we ran through our test, which was 100 different sequence iterations, and look at all the chromosomes and genomes that it found each sequence in. A nice, easy method of collecting this data, we can copy it directly from the site into something such as Excel. In addition to looking at the data on the website, we implemented our application as a standalone jar, which can be run through a command line interface or through a Node.js wrapper that was created. To run the Node.js wrapper, we have our Node.js file, which calls enhanced BLAT with a particular command or sequence. So let's take one sequence, for example, and run it through with that one sequence. Once it executes that jar, it will save the data as a JSON file. However, because this is in Node.js, we can actually add this to any pre-existing software such as OWL 2.0.
Our system can do and how it can run multiple jobs very quickly and need less user interaction. What do you think about what you saw?
It seems to work pretty efficiently. It seems like it'll cut down on the total amount of time I will need. Being able to walk away and let it run while I do something else is very advantageous in the lab, and I think it will actually be applicable to a lot of different people, either if they're designing a lot of different qPCR primers or probes, or for other applications, other people doing FISH or CHIRP or other methods where you need to make sure there's no off-target binding. So I think it'll be very useful.
Thank you so much, Christine, and I hope you can get use out of our project.
Thanks.